So I'd like to talk about phylogenetic models to understand avian influenza outbreaks and SARS-CoV-2. I will explain what phylogenetic models are in the next slide. So, to use pathogen phylogenetics, this is where you're using the sequence data of the pathogen and you're trying to use it to understand how the pathogen is spreading around the population. On the top left there, you have a phylogenetic tree. This phylogenetic tree is in fact of avian influenza, and the coloured dots are the sequences. The coloured dots are coloured according to year. In the middle panel, what you have is a molecular clock signal of those sequences. You can see that you've got this gradual accumulation of mutations over time. So the vertical axis is the genetic distance from the source point, and the horizontal axis is the time. And you've got this gradual accumulations of mutations over time according to a certain rate. You can go further. You can also include space upon the phylogenetic tree and model it together with the sequences, how they're evolving over time. And this lets you show the spreading pattern. So the picture on the right there, this is a map of avian influenza spreading. In fact, this is one of the earlier outbreaks. I will talk a bit more about the later outbreaks in a moment. And the coloured arrows are corresponding to the year in which that transmission occurred. This technique, though, is very powerful because you can use it for looking at the evolution and epidemiology of all kinds of animal and human viruses and bacteria. Because you're adding extra information as well as the sequences and modelling them together, you can look at things like cross-species transmissions, host adaptations, and interactions of the virus with the host. Because you're combining these evolutionary and spatial models, that lets you look at the sources and the roots and also the speed of the spread of the virus. Furthermore, you can also look at what is driving what that route is and what that speed is. So, as I mentioned, this is useful for all manner of pathogens, including influenza, avian influenza, BVD, SARS-CoV-2, PRRS, and bovine tuberculosis. What you need to do to make these models is you need virus sequence data, number one. Secondly, you need virus sequence data over time. So you need to have samples, and they need to be spread out over time. The other thing you need, and that you're getting from the sequences, is this concept of viral diversity or viral effective population size over time. And this might go up or down. And if it's going up, then this is the epidemic is growing. And in fact, what you're calculating there is the epidemic growth rate, or the R0 or the effective R. And you're calculating that from the virus sequences. You can go further yet again. So in the first case, you're imagining virus spreading in one population, but in fact, it's not one population. You have multiple types of population. You have multiple host species interacting. You have different types of avian species interacting for avian influenza. You have animal to human transmissions. And so when you're looking at the population scale, it's not just one population, and you're sampling sequences from individual hosts, and what you're sampling is you're assuming that you have one sequence per host, but those hosts might be spread amongst this kind of structured multi-level populations. You can also use this technique, by the way, on a much smaller scale as well. So you can use it also for transmission experiments. In that case, you're getting one sequence literally per individual or per pen, or sometimes you're getting multiple sequences per individual. So to use these sequences, so you make the phylogenetic trees, and then you can add on to the phylogenetic trees the other traits. And a trait could be a thing like a host type, so human, animal, different types of bird species, or it could be a continuous trait, so for example, latitude and longitude. And in the first instance, what you might assume is that you make the tree and then you map the traits onto the tree and you look to see how these are changing on the tree. So you have the picture on the top right there, a phylogenetic tree. It's an avian influenza tree. It's coloured by bird type. So we have the red domestic ducks at the top, which are being transmitted to the blue migrating wild birds, which are then being transmitted finally to the chickens in a kind of chicken spillover event. And the picture underneath, so I'm colouring the tree, but the reason I'm colouring the tree is so that you can see what the inferred host population was for that transmission pattern. In fact, this is a mathematical model, and the picture at the bottom is a rate matrix of the mathematical model for how quick you're going, on average, across the whole tree, from the wild migrating birds, which are blue, to the wild other birds, which are purple, being transmitted to the red domestic ducks or the green domestic chickens. So that's the simplest case. The next most simplest case is you say, okay, so now perhaps actually my traits might affect the tree. So we can model this as well. And this is where you might have the case where you have a mutation which increases the pathogenicity. So this is going to change the viral transmission properties, and this will therefore affect the tree. So I'll speak a little bit about high-path bird flu. This is from our most recent outbreaks. So first of all, this is high-path avian influenza H5N-X, and you'll see why in a minute. The X stands for any subtype. So from the UK, we have around 45 sequences publicly available so far. We have some from Scotland, we have some from Wales, and we have some from England. And the map shows roughly where they are. The colours on the map are according to subtype in the last 2 years. So it's mostly the purple H5N8s, and then some of the red H5N1s, and then a smattering of other subtypes. To understand how this comes about, so this is a tree from bird flu sequences for the last couple of years. It's from Europe and Asia, and also the UK. And in fact, many of these sequences are collected by our European partners, including TIS. And so what you see, first of all, it's a timescale tree, is that mostly you've got H5N8 in 2020 and early 21. And then secondly, you're seeing now, in this most recent season, you're seeing the clade at the bottom, which is H5N1. So what has happened is you've had a reassortment from H5N8, and you've changed the neuraminidase to be an N1. And so the picture at the very bottom is a cut through the tree to show you what is the dominant subtype. So you can see that it was actually back in mid-2020 that the H5N1s started, and then gradually they've been taking over. So now in this season, you've got mostly H5N1. You'll see a few other colours in there as well. This is because, in fact, as T said, you have it with this very particular clade, you have lots of reassortment with kind of resident low path, and so this strain picks up these other flu segments from the otherwise circulating low path. So this is mapping the tree in time and space. The colours are by subtype. It starts off in Africa, and now it's coming over to Europe as the purple H5N8, and then again it comes back over again as the red H5N1. So this is now repeating. And what you're seeing is a kind of quiet period and then like an explosion of H5N8 coming to Europe, and then again a reassortment and an explosion of H5N1 into Europe. And if you look closely, you can see as well there's a few other small reassortments and transmission of other subtypes. I'll just zoom in a little bit as well. So you've got this incursion to Europe, you've got this local mixing, and then now you'll have this other red H5N1 incursion. This is the view from the main surface protein, which is the hemagglutinin only. In fact, what it looks to me like it's happening is that you've got transmission of high-path bird flu, in fact, potentially on more than one flyway, and you have this mixing with the low path, so it's a very complicated dynamics. If we look just to the UK, you will see that in 2020-21, then it was mostly the H5N8 that came over to the UK, and you can see that for the most part it's coming in the European kind of direction, and it's kind of October, November, apart from the odd few which seem to be coming exceptionally from somewhere else. And then again you see in 2021 and then to the current that actually you've got this H5N1 coming over. But you can do more. So you see all these diffusion rates and all these diffusion patterns, so you're going to try and make sense of them. And so what can you do? Well, you can use a thing called the phylogenetic generalized linear model. And there's two forms of this. One form will work with a discrete trait. So this could be a discrete location, which could be like a country, or it could be a host type. And there, what you're saying is you're calculating the rate of going from one thing to another thing on the tree, and you're saying, well, okay, that is a rate matrix. What are the underlying predictors of this rate matrix? So this is what are the other underlying sets of matrices which could predict that matrix. In the second way, you could use a continuous trait, so you can use the latitude and longitude, and there what you're doing is you have a path through space that you've inferred from your tree, and you're trying to correlate that path through space with a path through an underlying predictor. So what types of things might you see? Well, we're trying to make this calculations. And so we used several sets of potential predictors to see which ones would be the most important. And these are things like, we in fact had risk factors across 10 different groups. We had biodiversity risk factors, bird flyway risk factors, climatic elevation, forest, land use, socioeconomic, vegetation, and water cover. And all these risk factors, they're essentially kind of gridded maps, right? So they're giant matrices, and you're trying to see which one of those explains the thing you're calculating from your sequences. And what we found was, first of all, of that particular data, that avian influenza tended to remain in or leave from areas with high poultry and human density. Well, that's not exactly a huge surprise, seeing as it's a poultry pathogen. But we also found other significant predictors in this, which were the flyways of the anseriforms, that's the ducks and the geese, and the passeriforms. Vegetation cover was also important, cropland use was important, urban land use was important, and tree cover, and of course, wetlands. All of these things add together quantitatively, and we are now in the process of unpicking what those things are to make our quantitative models. This method is the same method that we're going to try to use for the bovine viral diarrhea as well, from the earlier question. So secondly, I'd like to talk very briefly about phylogenetic analysis of SARS-CoV-2 sequences coming into Scotland. This is actually the same type of techniques as we've just seen for avian influenza. The first thing to say is that there is now a lot of full genome sequence data. It's been shared by GISAID and also in COG UK. And just to say, it's approximately 8 million sequences now. There's a lot of sequences. And in fact, the COG UK consortium is responsible for generating about a quarter of the global publicly available sequences. So, and then within Scotland, it's about 10% of the whole of the UK, which is proportionate to the number of people. So what can you use these sequences for? Well, you can track your important mutations and lineages, as I'm sure you've all heard about. Also, you have sufficient differences to infer clustering and transmission patterns, again, as we've been seeing with avian influenza, as we've been seeing with bovine viral diarrhea. And the types of questions you want to ask is, well, what are the infections between the cities and the regions, and is the epidemic growing and reducing? Again, this sequence data is well placed to answer these kinds of questions. So what you do is you have an entire massive global tree, which you need to split up into lineages. In fact, you also need to subsample and split the tree. You timescale these small subtrees and you estimate the growth rates. You add the discrete traits, in this case, Scotland, England, Wales, continents, or you can break it down further into health boards of Scotland, and then you calculate the imports from the other place into Scotland, and you record the time and the places on the branch of the trees, and you do this for all of the trees. So when we first did this, this is early data, this is 2020 data, this is just wave 1 and 2. We were able to show on the left there, that coloured bar graph is the imports into Scotland, and bear in mind this data was cut off in September 2020, so that's why it sort of disappears down at the end. But what you see at the start there is the imports into Scotland from England, which is the red colour, and Europe, which is the green colour, and then it kind of dies down during the lockdown, and then it maybe starts to increase a little bit. And then on the right there, you've got a snapshot of the imports into Scotland according to the health boards. Moving on, in Scotland, we have many lineages and variants. In fact, they've approximately come in these kind of waves. So the pale blue ones are the ones you were seeing in the previous slide, and then you have the B1177, which is September to December 2020, and then you have the Alpha, then you have the Delta, then you have the Omicron. You can use these and estimate the imports into Scotland again with these waves. Here the pink colour is England and the green is Europe. You can calculate the growth rates. And here's calculating the growth rates on the wave 2-3 sequences. And the colours on the left there, so you've got the timescale on the bottom, and the colours on the left are according to the mutations in the lineages. And yes, what you find is the alpha lineage that has a higher growth rate than the other lineages in this particular time period. And you can go further, you can look at the invasion of Delta into Scotland. And so what you're seeing there is coming in from just England, Wales, or Northern Ireland. I didn't do the other countries. And then the spread around Scotland. And then the background colours are kind of a indication of like the intensity of transmission within the health boards. And so I would say also with these growth rates, you have lineages and they have certain growth rates. What factors would affect those growth rates? Well, you've got your underlying population factors, your age, for example, movements, your vaccination immunity, and this goes for humans, the same as birds, the same as other farm animals. But you might have an intrinsic effect on the virus growth rate as well, like the mutations of the virus might actually be making the virus more transmissible. This might be what's happening with the H5N1 taking over from the H5N8. So to try to understand that, what you really have to do is integrate this experimental data in together with it as well. And there are experimental techniques which can tell you what in relative terms the fitness of one virus is compared to another virus if it contains a certain mutation. Some of these experiments are done for SARS-CoV-2 already. Some of these experiments are also being done for avian influenza as well. And so the challenge now is really to integrate this experimental intrinsic virus fitness effects with these population scale growth rates in order to be able to work out which lineage might be winning out in the end. So just in summary, you can use sequence data and these phylogenetic models to track incursions and spread in these structured multi-host type populations. You can use them for early warning if you can see the sequences from other countries. You can potentially make predictions or forecasts for spread in viral populations. You obviously need to know where are the current infections. You need to know the spreading patterns in the population. You need to understand the fitness of the new variants. And then once you have that, you can put these together in simulator and scenario generation models to try to at least have some sets of predictions of what would be coming. So with that, I have 1 million people to thank. I have people from EPIC and the University of Edinburgh. I have people from the Global Consortium for H5N8 and related viruses, which also include TIS. And I have people from the SARS-CoV-2 work within Scotland and COG UK, and I would like to give particular thanks to Lulu, who has been involved in both aspects of the work. Thank you very much.
Darren Shaw, University of Edinburgh. So in terms of prediction, I can see from your movie that it looks like it goes central belt and then elsewhere in Scotland. But in the last 2 years, we have had a really nice natural experiment in that different regions of Scotland were locked down to different degrees at various times. Are you, with your technique, able to see the impact of that going through time?
Not currently. However, I'm working with Public Health Scotland to answer exactly that question as we speak. So, and to answer more fully, yes, I think seeing as we have now enough time depth, like there's the 2 years and enough different things that have happened, and we certainly have enough sequences, there is the signal there to be seen.
Thank you very much. That was very interesting. Early on, I think it was the first slide, you showed the relationship between genetic distance and time. And I thought it looked remarkably linear. And I was thinking that it's remarkable because in macroevolution, there is evidence of punctuated equilibrium. And I just wondered whether this is a typical relationship and generalizable, and if it is, whether there's an explanation for the difference between the genetic shorter scale evolution and the macroevolution trends. So
first of all, the exact picture I was showing was for avian influenza. So flu viruses, particular clades of FMD, and now as we're seeing SARS-CoV-2, have this very strong molecular clock signal. HIV also, these have really good particular strong molecular clock signals. And all of these have in common that they are RNA viruses. So you have this quick accumulation of mutations. I think when you look at the macro scale, and also if you look at different serotypes of FMD as well, then because things are happening so quickly, you can see it in a sample spanning 10 years, 20 years, say, because this is actually many, many virus generation times, even if you're going from one host to another host, you can see that. But when things start to speciate and you have kind of different clusters of different virus diversities, then if you just looked at your sampling frame, you would lose that signal. So I think in the macro case, or rather in my mind, even if you have samples going back 100 years for hosts of like host genomes, this actually isn't much in evolutionary time, so you can't see that in the same way.
Just wondering if it makes sense to maybe approach the retracing of these trees in terms of an agent-based modeling.
Very much so. So what I'm hoping is that fundamentally you're calculating the rate of one thing going to another thing according to the phylogenies, but those rates could very easily be used into an agent-based model.
Thanks very much, Sam. It was great. Dominic Mellor, University of Glasgow. I'm a bit concerned about the focus just on pathogens. I can't believe that the sort of variation in disease isn't affected to some extent by the microbiome that happens to be at home in various hosts. And I don't know that we're really giving it much attention at all. It may not be that something that can fit into your approach, but would you comment on that?
So I think on the one hand, my approach is using sequences from pathogens. So, but on the other hand, you are trying to understand what are the host statuses which would make the virus more or less transmissible, and also the effect on the host. So I think in my approach, what I would try to do is have some metric of the microbiomes of the hosts and incorporate that into the phylogenetic GLM. So I think the short answer to this is it's not currently done, but it potentially is a possibility. And I agree, I think because the microbiome can have such an effect on the host immune status that really, in the end, if we could include it, this would be very good.